Welcome everyone to the third session of the spring series focusing on aging. It's with great pleasure that we welcome to speak to us again Wayne. We're going to ask Alistair, our Director of Science, to introduce Wayne. Alistair, over to you and then over to Wayne.
Okay, thank you, Jeffrey. My name's Alistair. I'm the Director of Science. And before we get to the main event of Wayne, I'm just going to provide a little bit of background and some interesting thoughts, I hope, which will get people thinking. Probably the first one is we're talking about the ATPase today, is that we're all very familiar with this diagram on the left, which is the electron transport chain and the development of the proton gradient and this ATP synthase on the right. But what's really quite interesting, of course, is this is actually a rotary motor. And when those biologists amongst us will go, well, that's actually really quite interesting, isn't it? Because, how can things rotate in biology? And of course, it shares a lot with the what we might see as a turbine generator, which is this thing in the middle. But of course, it's a really interesting story, and maybe one not everybody's thought about, of course. But where did it come from? And one of our favourite theories on the origins of life is around the thermal vent. And I will point out other theories on the origins of life are available, but we stick with this one, because certainly for me, it seems to be one of the most logical. And this is that on the very early Earth, and as indeed there are today, there are what we call alkaline thermal vents. And this is where the geology of the Earth has produced hydrogen, and it upwells into the water. In particular, in the early Earth, was acidic seawater. And this provides a very big energy gradient. And the theory being that actually, because of thermodynamics and auto-organization and dissipation, you get the formation of structures which are basically come into being to try and dissipate this energy potential. And this leads to structure, and this is the idea behind life actually started. And certainly there's a lot of evidence now and some good theory suggesting this is entirely possible, and these thermal vents can generate most of the early chemistry and the chemistry leading to life we know nowadays. And in particular, it can generate things like membrane, or the fatty acids and membranes, and with components like iron sulfides and things, you get all the components you need. Now, some of the theory is in the early days, there was an iron gradient, but when you start going into the literature, there's certainly evidence from people, and this including from D'Souza, but also others, that genetically, it seems that this rotary component came in very early in life, possibly even before life left the thermal vent. Now, again, this is all speculation to some degree, but it does make a rather interesting story. And obviously today, we know all of this, of course, within the mitochondria, but this could have started right at the beginning of life, which is quite interesting. And one of the interesting things is when you start to think about this dissipating concept, and I think we've talked about this before, of course, early life was probably very, very inefficient in the sense that there was plenty of energy, and how it dissipated the energy in the proton gradient was to make more compounds. It turned high energy stuff into low energy compounds. But today, and this is something, again, which not everybody appreciates, life is actually fairly inefficient in the sense that it seems to waste 20 to 40% of the energy that it's capturing. And everybody knows who's into mitochondrial research knows about uncoupling. And this is a process whereby the proton gradient, which is generated, is effectively short-circuited. But it seems to be very, very important in controlling life. And in effect, kind of short-circuits the electrical system. But of course, it's fundamental to dissipation and self-organization, and so this concept of flux stability comes into this. But it also seems to be important in developing adaptability. And when you look at certainly the mitochondria, but indeed prokaryotes, they have this gradient around them, which of course senses and changes and adapts to any shift in the environment. And this, of course, is generated by producing an ion gradient. And the F1 ATPase, which Wayne's going to talk about a lot in much more detail, is clearly very important in this process, and certainly can operate in reverse. And not everybody thinks it operates, it doesn't just produce ATP, it can use it to maintain the gradient. And this is all very tightly integrated with controlling redox, and back to our electrical circuit again. And there's evidence perhaps the ATPase itself is important in this process. And then again, Wayne will talk about this potentially. Mitochondria are also quite warm, and this goes back to the dissipation idea. Now, there are some people who say this isn't possible, but there's quite recent papers suggesting that mitochondria are operating almost 10 to 15 degrees hotter than their surroundings. Now, this hasn't been proved by other groups yet, but it's an interesting idea in terms of thermodynamics. So the key message really here is that the energy flux itself results in structure. And we know that maintaining structure is a key in the rate of aging. one of the oldest, in terms of evolution, aspects to aging is a loss of protein metabolism and what they call proteostasis, i.e. proteins fall to pieces and they're not replaced. And that seems to be one of the earliest things, but of course it's associated with lots of other things like genomic instability, mitochondrial dysfunction, change in epigenetics. But the most ancient is this idea to maintain protein. And it kind of suggests that there is obviously underlying life. There's a requirement for an energy flux to maintain structure. And when we start to think about movement, and of course we're talking about exercise and aging today, it would seem that movement is another mechanism to maintain dissipation. And some species, for instance humans, but other animals and other creatures which move around, have probably become, through evolution, dependent on this movement to maintain their structure to some degree. They can survive without moving, but they're not as good unless they move. And it seems to be very tightly linked into their ability to be robust and adapt to changing situations. And so currently for humans, it seems to suggest that we have to maintain a stimulus of movement to optimize our systems. And this, of course, includes mitochondria and the turbines, the ATPase, which we talk about. Okay, so the need for physical activity. Well, certainly, I'm actually I am a qualified a gym instructor, but that's beside the point. But I've had this drilled into me for a long time. But certainly the evidence is that activity and the importance of physical activity and exercise, which is kind of more prescribed physical activity, has been known to be important for health for thousands of years. And nowadays it's become recognized as a medicine, possibly one of the best medicines ever. It's defined as any body movement that results in energy expenditure above that of resting. And it's most normally measured in metabolic equivalent tasks, which is what we call METs. And so light exercise or light physical activity is defined as anything above 3. So that's 3 times what you might be doing resting, moderate 3 to 6, and vigorous to 6. But when we talk about elite athletes, these are folk who can go up to 10 to 15 times their resting rate. And it's an enormous increase in metabolic capacity and ability to maintain it. And we know it's anti-inflammatory, it's senolytic, exercise results in the removal of damaged organelles and cells, and it modulates all the pathways we know in the aging process. And this is NRF2, FOXO, AMPK, mTOR, many others, probably P53, there's all sorts of other things involved in there. And as you said, it seems to enhance mitochondrial function, but it certainly seems to make one more robust. And I say this slows aging compared to normal. We've got to remember what we measure as normal is not necessarily healthy, because of course we're living in a large variable population. What is the proof? And I've pinched this from a couple of more recent publications. This is a graph showing the cardiorespiratory fitness related to mortality. And it shows very clearly, if you put people on a treadmill and measure their fitness, their mortality is inverted to how fit they are. The fitter you are, the lower your chances of mortality. And the interesting thing, and this is a debate that's been going on amongst the exercise folk for years and years and years, is there an upper limit to how much exercise results in benefit? And the answer is not as much as we think. We don't quite know where the top level is. For most people, we get nowhere near it. Maybe running down Africa, as that chap really did, running a couple of marathons a day probably is too much. But for many people, we never reach the top limit, which is, I find, quite interesting. And this is from a very large study, but many other studies support the same thing. But of course, cardiorespiratory fitness requires a good, healthy mitochondria and, of course, good, healthy ATPase. And we know that exercise, one of the things it does is stimulate the turnover and increase of healthy mitochondria, the best way of putting it, and our ability to resist oxidative stress. And this is a much earlier study where they simply looked at the same thing, but from the MET's point of view, and again showed that people who moved around a lot more tended to have a lower risk of mortality and increased their number of years of life. And interestingly, exercise has benefit right across the age range. Even the very elderly people, if they can move, it does seem to help. And the key mechanism, of course, which we're very interested in, involves the generation of ROS and adaptation, which is some would call hormesis. But this idea of what we call mitohormesis has been around a very long time and involves many of the key players. But the key thing here is stressing the mitochondria results in an adaptive response, which seems to unblind is very important in exercise. And so this is very much the standard kind of diagram that everybody's seen. But I love this quote by Dubansky, which is, nothing in biology makes sense except in light of evolution. We could not have evolved without adaptation. If anybody argues that this is important, it's extremely important, because if we didn't have stress, we wouldn't have adapted or evolved, as we clearly have. And certainly we've always said that, there's always been a saying, hormesis, what doesn't kill you makes you stronger, but we've sort of added to it, makes you smarter and longer lived. And you've probably seen this diagram before, but this is the basis of it. And this is where we've basically looked at the data and said, well, if everybody lived a really healthy lifestyle, how long are they likely to live? The evidence could be closer to 100. And the most important bit, in which Jimmy's very, very keen on, is that living a healthy lifestyle, assuming everything goes well for you, you experience morbidity compression. And this is that you live most of your life very healthily. You have a good health span and everything goes wrong right at the end. Whereas if you live a really bad lifestyle, the opposite happens. You don't live as long and you spend a lot greater part of your life in ill health. This is it's a very clear message, I think we've seen. The blue lines here just represent the population average. And certainly we were thinking about this a long time ago, and this is diagrams from a paper we published several years ago, many years ago actually, when we were very interested in the metabolic syndrome. And for those of you who are familiar with this or don't familiar with this, this is a syndrome which seems to be associated with the poor lifestyle. You have increased tummy fat, and this is what they call visceral adipose tissue. You get increasing insulin resistance, and of course this predisposes to diabetes. You also get high blood pressure, your lipids go all funny. For instance, your good cholesterol, the HDL, drops, and it's associated with inflammation and mitochondrial dysfunction. What we know is as soon as you start to exercise, in particular, your visceral adipose tissue volume drops very quickly. So your body adapts quite quickly to this. But the key thing here, and this is, we've kind of mentioned this again before, is this exercising seems to increase your metabolic flight envelope, in a sense, your metabolic flexibility. You become more able to resist things. In particular, for instance, if you have to suddenly run or resist, in particular, say, a virus. And again, this is this idea that if you're metabolically healthy, and this is associated with fitness and obviously eating reasonably well, it seems that you're more able to resist viruses. And as we now know, many viruses, including our friend COVID-19, modulates mitochondrial function as part of how it results in its own replication and infection. And the evidence is building quite strongly now, and we suggest this, as did many other people, that we have to look at mitochondrial health in COVID, is that people who are generally very fit and aren't suffering from comorbidities, and certainly things like the metabolic syndrome, fare much better. And the data is beginning to support this quite well now. There's another factor which isn't always completely appreciated, and which comes out of all of this, and this comes back to the exercise idea, and this is your muscle to fat ratio. And certainly, if you exercise a lot, you tend to have more active muscle, and we know that muscle generally has an anti-inflammatory balance in your body. But adipose tissue, if you have too much, it becomes inflammatory. And certainly it would suggest that if you exercise regularly, you get lots of healthy muscle, which is anti-inflammatory, and less inflammatory adipose tissue. Very simple idea, but potentially tells us a great deal about how we might lead to a healthier life. But of course, we're all going down to where does this go with when we start to think about the ATPase. Now, one of the things that we've certainly been interested in for a very long time is mitochondria. And I certainly grew up, and certainly in my biochemistry days, thinking that mitochondria were these things that are, well, the necessary evil. They produce ATP, but they also produce a lot of reactive oxygen species, and they probably cause aging. But the truth is, and it's becoming very clear now, they modulate ROS very tightly indeed, to the point that they can actually act as net sinks of ROS. They can mop up hydrogen peroxide, so they have their own very powerful antioxidant systems. And certainly this idea, which was raised by Aon and colleagues, was a long time ago, is that mitochondria operate in a certain, like a U-shaped curve, where too much or too little of their activity is damaging. But it seems they've evolved to operate in a Goldilocks zone, where they rely on having to be utilized quite heavily. And that's where they produce the least ROS. If you want to look, this is the paper, if you want to have a look at it. And as we know, ATPase defects certainly can lead to increased ROS in the cell, and ATPase is certainly therefore, because of its role, very important in controlling redox. And I think there's quite a lot of evidence it's modulated by redox directly, but also, and this is the interesting thing, of course, it does operate in both directions. And as it now turns out, it probably operates in both directions within the same mitochondria, in a healthy mitochondria. Suggesting, as I'm sure Wayne will talk about, cristae within the mitochondria, of course, they're all in different places at different stages of what they're doing. So it's a very, very flexible molecule, unit, subunit, and function. Now, this is sorry, I'll just digress slightly here, but I always thought this is quite an interesting way of showing this. This is a very old diagram, basically showing electron flow, because this is related obviously to the proton gradient and the ATPase. If we think about acute inflammation, one of the key things that happens is something gets damaged, or we say we get hypoxia or something, and we get a generation of reactive oxygen signal. This is partly how the body adapts. And one of the most important things that happens, of course, is it's self-limiting, or it should be self-limiting, because it induces a negative feedback spiral involving antioxidants and eventually turnover of more mitochondria and removing the damaged ones. And in theory, it becomes self-resolving, so your inflammation disappears. But if we think about chronic inflammation, what seems to happen is that for some reason, the negative feedback cycle doesn't seem to operate as well as it should do. And this is certainly what seems to happen partly in the aging process. We end up with subclinical inflammation. And it can lead to damage and like a vicious spiral. This certainly happens in COVID, we think. There's more evidence for this now. But when we come to exercise, which is interesting, again, you get a similar kind of thing. Exercise is inflammatory, or at least initially it is. It damages your muscles, they all get a bit tears in them, they get a bit stressed, but almost straight away they start to generate this feedback mechanism, which is trying to reduce the reactive oxygen species and the damage. There's a big signal, but it comes back and tries to sort itself out, and eventually reduces the inflammation. But of course, what is different from chronic inflammation is, of course, that this process doesn't stop. And here we get the adaptability. And as we go through this, one of the things that happens is, of course, you increase your metabolic flexibility and capacity. And this goes hand in hand with the ability to control oxidative stress. And this is really key. And this is why one of the reasons exercise is so important. And the other important aspect here, of course, everybody just thinks that, oh, it's just in the muscles, isn't it? Well, the answer is no, it isn't, because mitochondria talk to every other mitochondria in the body, and they do this through metabolites, myokines, extracellular vesicles, probably RNA, and probably things we haven't even thought of yet. And so they interact with every other component in the body, and this induces metabolic flexibility right through the body. So and this system seems to be very tightly integrated. Way I like to look at this, sorry, this is my favorite little diagram, is, and I've used this a long time ago, actually, when we were talking to people in Saudi, actually, is how do you explain this metabolic engine idea? And one of the ways we use this is to think about a car. Now, here's your average car. Sorry, it's a little cartoon I picked off the internet. If you leave it in the garage, it turns into a Trabant. Now, those of you who've driven a Trabant, and I have, it's an interesting experience. I recommend it. It's quite fun. Very low powered. It's not terribly reliable, fairly inefficient. It's got no energy recapture. But if you regularly drive it, it could turn into a Formula One car. And of course, you're looking at a completely different beast here. And this is an animal which is 1,000 horsepower, 15,000 RPM, 6 cylinders, etc. And so there's this difference. If we don't use it, we lose it. If we use it in the right way, we actually enhance its function. So just coming towards the end. So there's some interesting things here about exercise, ATP synthase, and there's certainly some questions. Is more healthy functional ATPase equals better metabolic flexibility? I think Wayne should be able to answer this one. Why did the turbine evolve? And this is, I think, a fascinating question. Was it the simplest solution to dissipating the energy gradient and vorticity? And this goes back to other aspects which we discussed in various lectures, including spin and the role of chirality. Did movement, and here's a thing which I, again, just think about this. Did movement evolve to dissipate energy gradients, which was enabled by something like the ATPase, so leading to a feed-forward cycle and reinforcement of a dissipating structure, i.e. the structures which enable biology to survive. So for some creatures, is movement essential to maintain structure at the most basic level? Without movement, it starts to fall to pieces. Anyway, I'll leave you with that, and I'll now introduce Wayne. Wayne's from Arizona State University. He holds professorships in several different departments at Arizona. He's an advisor to us at the Guy Foundation, and we met him at the Gordon Conference in bioenergetics, where there was lots of very interesting discussions going on in mitochondria, in which he was the chair, and he was very helpful there. And he now works with Mike Levine and putting ATPase into subunits and nanobots. This is another fascinating area, but we're not talking about that today. His key focus has been on the F1FO ATPase and how to visualize it. He's got some fantastic videos of rotating nanorods. They're absolutely amazing. He holds many patents. He's got over 1,800 citations and an H index of 23. And today, I think he's going to talk about ATPase and the aging mitochondria. So with that, I shall stop and hand over to Wayne.
Yes, let me start out and talk about this rather depressing subject of how we all age and how this affects, is driven by our mitochondria in part. So over on the left, you'll see an electron micrograph of a cross-section of a mitochondria, and this is a cartoon that shows how it has an outer membrane and then an inner membrane, and the most of the energy protein complexes that are associated with making ATP are located on this inner membrane, but it uses these spaces, the matrix and the inner membrane space, to drive the synthesis of ATP. So it starts out by you're eating food. You can either eat fat. I mean, this is methane. It's a long, hydrocarbon chain would be equivalent. Or you can eat a carbohydrate, and that starts getting processed in the cytoplasm of a cell, and these get oxidized. If you start with a carbohydrate, well, here's alcohol, but you can, there are stepwise oxidation events, and it's called oxidation because anytime you interact with oxygen, then oxygen is very electron withdrawing, so it doesn't share electrons well, so it actually will pull the electrons away from the carbon and oxidizes it, so it has lower energy once it's oxidized. So the more covalent bonds you have with oxygen, this is the carbon has 1, 2, 3 bonds with oxygen. Finally, 4, CO2 is the most oxidized state. Well, once it gets to a certain stage of this, it moves inside to the matrix of the mitochondria, where it ultimately goes to carbon dioxide. And in so doing, the enzymes there are generating the reduced form of NADH. So it gains an electron, and that way the positive charge is reduced. That's why it's called that. And to balance that charge, then you add a proton. So then it gets oxidized, it loses 2 electrons, and now it's NAD+. Whenever it's oxidized, it has less energy than when it's reduced. And the electrons keep going down energy-wise until finally it reduces oxygen to water. In so doing, it pumps protons, and I'll go to the next figure here. This is now we're showing that we have the structures of all these components where you're oxidizing the NADH and you're pumping protons through these pumps. You're also reducing ubiquinone. You can buy ubiquinone in one of the health food stores, but it really won't help you very much because your body metabolizes it. Nevertheless, it shuttles the electrons from complex 1 to complex 3. And you can also shuttle it from complex 2 to complex 3. Ultimately, the electrons get transferred to a small molecule protein called cytochrome C that moves the electrons over to complex 4, where oxygen then is reduced. I bring this up because cytochrome C not only works as an important shuttle here for electrons, but it also is a trigger of apoptosis, programmed cell death, that says, hey, this cell is done, it's done its work, and it's time for it to go. So in the meantime, all these protons are accumulating on one side of the membrane. In addition, you're having a difference in charge across the membrane that ends up being a membrane potential. The protons can specifically go through a pore over on the F1FO complex. F1 is the part that's sticking out of the membrane, and it is responsible for making ATP. FO is the part where the protons go through and drive the rotation of this c-subunit ring, a rotor, relative to the stator that's actually not shown here. There's another thing to consider, and that is that this sometimes does get backed up, and when it does, then you can create free radicals like a single, this is ubiquinone accepts 2 electrons that's compensated by binding two protons on this side of the membrane, and when it delivers them to the other side of the membrane. But if you get a single electron, it can form a free radical that then can react with all the oxygen that's present and form these oxygen radicals that you have, superoxide and hydrogen peroxide, et cetera. So that this is a very destructive thing every time you have a free radical. It wants to react immediately with everything. Okay, so where did the mitochondria come from? Well, it's just recently been determined that the organism that gave rise to eukaryotes was an ASGARD archaebacterium. And there's an electron micrograph of it. And so it probably had very low ATP synthesis activity. It had an A-type ATP synthase. And so in order to gain efficiency, it swallowed an alpha-proteobacterium that had an F-type ATP synthase. And when it did, ultimately, the alpha-proteobacterium became the mitochondria and started to make ATP much more efficiently than the ASGARD was able to do. And after a lot of evolution, you have a whole internal membrane system where the mitochondria is one of these internal organelle components. Now, initially, you had the entire genome of this alpha proteobacterium, so it could recreate the entire organism. But over the years, there was such a good relationship between the two that most of the genes for the alpha proteobacterium moved over to the nucleus, so that only a few remain. Nevertheless, you can kind of consider a mitochondrion a bit like your pet cat. While, you may call it a pet, it still has an awful lot of independence, and it has its own mind. So in fact, it will replicate itself, and it's kind of living its own life within your cells. So this is the ultimate genome of, we know it today. There's only 37 genes in the mitochondria. And when it divides and replicates, and that encodes 13 proteins, including 22 tRNAs and 2 rRNAs. And here there's 2 subunits of all the different subunits of the F1FO. Only 2 are encoded by the mitochondrial genome, one of which is extremely important. This is, this is the A subunit, which is the stator that allows the protons to be transported across the membrane to drive rotation. So in today's world, we now have cryo-EM and its associated technology, cryo-electron tomography, where you can visualize 3 dimensions of organelles. And this is a recent paper by Werner Krulbrandt's group, where he's taken in situ mitochondria and visualized what the 3-dimensional structure is. And so you can now see in 3D what the organization of those internal membranes is. Now, membranes are a lipid bilayer, and they're fluid in 2 dimensions. They're not fluid across the membrane, but they are in the other 2 dimensions along the surface of the membrane. So it's very difficult to get these very tight curves that you have that are found in the mitochondria. In fact, if you take and look at a cross section of one of these are called cristae. And the cristae, in fact, in a very healthy mitochondrion, they're fenestrated. They have little windows in them, and that increases the amount of edges that you have for this curvature. In addition, you'll see that in a healthy mitochondrial, you'll have very small, tight junctions with the outer membrane. That's shown here, this being the outer membrane and this being part of the cristae. Well, Werner has just about a week or so ago shared a preprint of a recent paper he is in about in the process of publishing with me. It's a much higher resolution structure of the mitochondria in 3D, and it's from Polytomella, which is an algae. And so you can now see in detail where all the F1FOs are along the surface of the cristae. The F1FOs in mitochondria form dimers. I had previously just shown you what a monomer of this as being an F1, here's the FO, and here's the peripheral stalk. This all forms the stator, and this is the rotor that comes up through here to make ATP. So two of them are stuck together, and in fact, there are several different proteins that join these two together that define what the angle is between them. And that angle determines the curvature of the cristae. So when you have fenestration, then you have these, you're increasing the edges and they form these twisting things as a result. So you're increasing the number of F1FO complexes you have in the mitochondria relative to the electron transfer complexes. More ATP synthase, more ATP that you're getting, more efficiency that you're getting. So this was, I think it's quite remarkable in that you're seeing this as it was determined inside a cell. Okay, so as I say, mitochondria kind of live their own secret lives. In order to do that, they have to divide and reproduce. So typically in a human, a female is born with her eggs already there, so for the next generation, and in each egg there's about 5 mitochondria. In each of these mitochondria, there's about 2 to 10 copies of the mitochondrial DNA. So the mitochondria from the sperm very, very rarely ever make it into the fertilized egg, so that all of your mitochondrial and your mitochondrial DNA come from the female. And you can trace back your mother's heritage through your mother's family by looking at the DNA sequence of the mitochondria. So as you develop into an adult human, all your mitochondria come from those initial 5 mitochondria. And so it has to divide. That's a process called fission. And now it's known that fission requires a series of proteins that facilitate this in order to make this happen. And the mitochondria will associate with an endoplasmic reticulum in order to make this happen. And at the time, the mitochondrial DNA will align along the middle of the mitochondria in order so that as it replicates, then half the copies will go over here and half the copies will go over here. At the same time, damage occurs because this is high energy stuff and that's going on. And it's the energy is so high that the membrane potential, if you were to make it even higher, it would explode those inner membranes. People have forced it to that point. So it's, in addition, you have all the ability to create the reactive oxygen species, ROS, and so damage can occur. Also, there is things like UV light that can damage the mitochondrial DNA, and while there's only a small number of things that are proteins that are being made from it, they're incredibly important to make this all work. So one of the ways of getting around this is to have, if a mitochondria gets damaged, is to have it fuse. So as the mitochondria is swimming around in the cell, there will be fission going on, and especially as you exercise, you'll create more mitochondria, but there's also fusion. As a mitochondria gets damaged, then there are a whole group of proteins that will facilitate this fusion so that you can have one damaged and you can have one that is intact. And when they fuse, then you can recover the function of the one that was damaged. So keep in mind, as we go through this, that OPA1 is one of these proteins, and it'll keep coming up time and time again. So as Alistair was saying, that we all need a little bit of stress in our life to make things better, and mild mitochondrial perturbation is an important part of this that delays aging and age-related functional decline. So the fusion-dominated impacts, if you increase fusion, and there's some model organisms, mice, Drosophila, C. elegans, you'll see there's a couple other ones. So increased fusion in C. elegans and Drosophila increase longevity, the length of time that the cell will survive. And inhibition of fission also increases longevity, at least as measured in yeast. And there's a couple more processes that mediate the fusion process, and like TORC1, which is a very important mediator of a lot of cellular processes, and the AMPK, it'll also mediate longevity by requiring both fusion and fission. And so this is a hormesis effect. Alistair likes hormesis, where you have to have a balance of fusion and fission in order to extend lifestyle and lead a healthy life. In mice, caloric restriction will increase the mitochondrial length and width in muscle, and starvation increases the fission proteins like the DRP1. The fusion increases the efficiency, maintains ATP production, even when you're starving, which is sort of an important thing, that you need to maintain this when under starving conditions. So unopposed fission shortens your lifespan, and it's been shown clearly in yeast. And so this is often referred to as mitochondrial fragmentation. When you have fusion occurring, then that is developing a very healthy mitochondrial network, where the mitochondria are interacting with each other and restoring them function to damaged situations. So a lot of the studies of how this gets balanced has been to use organisms where you can knock out or knock in the various proteins that are involved with fission and fusion. And so here we have excess glucose increases ROS, and it also increases the DRP1 fission protein. Knockdown of OPA1, the fusion protein, reduces the mitochondrial membrane potential and increases the proton leak, uncoupling ATP synthesis. And I guess I should have made a bit more of a thing when I had the figure on there about this whole idea of coupling, that Peter Mitchell got the Nobel Prize back in the 1970s for identifying that the ATP synthase is completely a separate protein complex from the electron transfer. That was a big debated thing at the time. And how that works is that it's coupled through that proton and gradient, but it can be uncoupled. You can add a small molecule. One of the ones that's often used today is something called c-CCP, and c-CCP will rapidly shuttle the protons back across the membrane and short circuit the whole system, so that electron transfers, pumping protons wildly, but the protons never get to the ATP synthase. So actually, long before this whole coupling thing, so the two become uncoupled. You get lots of electron transfer, but no ATP. And this was first discovered, even though people didn't know what it was, back in World War I. In munitions factories, one of the compounds used to make munitions was uncoupling people's ATP synthase from their electron transfer, and they started losing weight. And so it was actually used as a weight loss drug for a while, people would get very hot and sweaty, as Alistair pointed out, that the mitochondria runs hot, but especially when it's uncoupled. And brown mitochondria that's in your back, that's how you maintain a body temperature, is because you mostly have uncoupled mitochondria in brown mitochondria that are in your body. Ultimately, people didn't understand how dangerous it was to use an uncoupler as a weight loss, and after many people died, it was outlawed. Okay, so one of the things that there are differences between unicellular and multicellular organism is that if you completely ablate fission and fusion, then it shortens the lifespan in yeast, but it increases the longevity in worms. I don't know that this has been explained how that happens. Okay, so now, I don't know how many of you may be familiar with the life cycle of a cell, but the top figure over here is showing the life cycle of a cell. And so a cell will ultimately end up dividing and forming two daughter cells. And in so doing, it goes through these various stages of mitosis. So you'll have let's start with the G2 stage, where the cell is actively growing. Then you'll have the mitotic process go, where you'll have the DNA will replicate, and ultimately then the two cells will split. Then there is another period of cell growth, followed by DNA synthesis, and the cycle repeats. Well, as it turns out, that the mitochondria, as I said, have to divide as well, so that there is an equal number of mitochondria that ends up in the daughter cells. And so if you, it's been shown now by manipulating these proteins that the DRP1, which is a fission protein, it'll cause the cell to enter into the S phase over here for DNA synthesis of the mitochondrial DNA, and the G2M phase right through this part. During that, the mitochondria tend to fragment to ensure the equal distribution between the daughter cells. When you knock down the Fis1 protein, the fission, then that causes the cells to get stuck in this G2M phase, so that they no longer will divide and stop growing. So this leads to cellular senescence. So the cells ultimately, many cells stop dividing. Nerve cells are well known for this, but of course, skin cells don't go into senescence. They're always at the base of your skin, they're always dividing, but ultimately they get to the outside and they have a lifespan where they will die and flake off. So you're constantly replacing them. So the cells in senescence stop dividing. They just go into this quiet state. Ultimately, they secrete inflammatory signaling factors that are a hallmark of aging. One of these is cytochrome C, that tells the cell, hey, you know, it's your time is up. And so anyway, you end up having mitochondrial biogenesis, ATP synthase decreases, and if you have an organism that is starting to age, like we all are, then the tissues accumulate a lot of senescent cells. And so they have reduced fission proteins and increased fusion protein levels. Senescence ends up associated with these hypertubular mitochondrial networks, and you've got to clear these cells in order to keep your life going. And so one of the ways to do this is by apoptosis. So the fusion enables the mitochondrial complex content to mix, facilitating the function and correct these mitochondrial deficits that occur by ROS or by DNA deficits that due to destruction of the DNA in one of the two mitochondria. And the fission keeps increasing the number of mitochondria that you have. and which will increase with the exercise. And so anyway, this ultimately segregates the dysfunctional and superfluous mitochondria to get rid of them. And this is a process called mitophagy, where you're actually getting rid of bad mitochondria. And so someone did a knock-in gene that would express a mutant of polymerase POLG gamma. So the polymerase will replicate the mitochondrial gene. And since you have multiple copies, then this is protecting you, because unless you, okay, well, if your mother is giving birth to you after age 35, because all those eggs have been sitting there all these years, once you get past 35, the mitochondrial genome starts to degrade. And so you'll have, of those 5 mitochondria, you'll start to have a lot more dysfunctional DNA that will give rise to mutated proteins that lose the ability to make ATP. So this becomes a major problem. The polymerase is very good at trying to keep the sequence of these genes as in the integrity as high as it can, the fidelity, but it does this by comparing what all the different sequences are of the multiple copies of the mitochondrial DNA. If you have a dominant number of mutated ones, it's going to say, well, that's the normal one, and it's going to replace those. Okay, so if you knock out, if you put in this proofreading deficient form of polymerase, you start to generate all sorts of mutations in your mitochondrial DNA. And so this just exhibits premature aging. The poor mice have hair loss and graying and sarcopenia and deafness. I mean, well, anyway, this is what happens. They develop mitochondrial myopathy in muscle and heart, which is decreased via fusion. So fusion will maintain the mitochondrial DNA levels and the mitochondrial DNA fidelity. Okay, so how does this happen? So there are a couple proteins involved with this. And so the mitophagy will occur mediated by these proteins called PINK and PARKIN, and they work with ubiquitin. And PARKIN is named because when you have mutated forms of PINK1 and PARKIN, it gives rise to familiar Parkinson's diseases. So it starts to accumulate on the mitochondrial surface and upon stress induced. And one of the first things you get is you depolarize the membrane potential of the inside membrane. So since ATP synthesis depends on this membrane potential, you're dramatically decreasing the ability to make ATP. And then it recruits these other proteins that actually ubiquinate the outer mitochondrial membrane, and it facilitates its removal. And there's another way to do this by receptor-mediated mitophagy. And ultimately, what happens is that you form this mitochondrial permeability transition pore. And that causes the inner membrane to swell, and then it bursts. That releases cytochrome C to induce apoptosis. Now, the mitochondrial permeability transition pore is not well defined, but in recent years, it has become associated. People think that it's due to the F1FO ATP synthase. Something happens to it. What happens to it as to date, we don't know. People have lots of ideas, but it's one of the big things to discover out there. There's another way that is a lot less invasive than actually having mitophagy, in that if you get oxidized proteins and lipids, then the bad parts can bleb off and they can be targeted over to the lysosome for disposal. And that way you can keep the mitochondria healthy without actually going through all this problem to do fission and fusion or have mitophagy occur. So ultimately, this is a balancing effect where if fusion and fission are both required, if fusion dominates, that it increases longevity and insulin sensitivity, glucose tolerance. And if you have fission that dominates, then you get premature aging, cardiomyopathy, obesity, diabetes. And this is promoted by starvation, whereas this is promoted by nutrient excess. You eat too much. So one of the deadly sins. And so anyway, in fact, insulin has a bimodal effect. It promotes fission, but that decreases longevity, but it also upregulates OPA1, which is a fusion protein that leads to a healthy interconnected mitochondrial network. And caloric restriction promotes a healthy mitochondrial network, and it's a very effective way to improve health and decelerate aging. So push that dessert away the next time you see it. All right, so now let me go back to Werner Kulbrandt and his aging related studies by looking at the three-dimensional structure of mitochondria. And he first did this in an organism, Podospora anserina. It's a fungus that's used as a model organism for aging. And one of the reasons you might say, wait a minute, how can this relate to humans? But it is, a eukaryote, single cell. It has a 15 to 20 day defined lifespan, so that it's easy to study. It's easy to get from when it's just young to very old, and you can follow this. And it shares a lot of age-related effects with mammals: generation, scavenging of oxygen radicals, mitochondrial DNA instability, protein quality control, and aging. And you knock out the DNM1, and that'll delay the mito network fragmentation, and it'll increase the lifespan of this fungus by 9-fold. And the OPA1 knockout, or at least the analogous protein in this fungus, will actually reduce the lifespan. And one further thing is that while the mitochondria permeability transition pore complex is thought to be, has thought to be strictly something that occurs in mammals, overexpressing cyclosporin D will actually affect this. And so it appears that this Podospora can in fact form an MPTP complex. So Kulbrandt's study, I think I jumped over, no I didn't. Kulbrandt's studies show that as you go from healthy to aging, one of the things that happens is that at the beginning, when it's a young Podospora, you have lots of nicely formed cristae, and you have your F1FO dimers. But as you progress towards to aging, you'll see that these proteins start to break down that are linking the dimers. Ultimately, they form monomers. And in so doing, you see the entire loss of the cristae inside the mitochondria. At the same time, the very latter stages, you'll start to see this increase in the an association of the inner membrane with the outer membrane. So this is an animation of what we think is going on here, that in your healthy young mitochondria, you have a nicely formed cristae, and where you have your dimers that are at the very end forming this curvature. At this end, you have proteins that are forming a cristae junction that are allowing this to be a separate space, aqueous space, from this space. And that allows the formation of a proton gradient and a membrane potential that is produced by the electron transfer complexes that fill in along the length here. As you start to have aging, you see that there's a breakdown here, and ultimately you're losing your cristae altogether, one of the final steps is that now you've gone from F1FO dimers and you've lost the entire space, and then finally you have a total collapse and the permeability transition pore forms and you're done. So there's a bit more to this, that the permeability transition pore is formed by, induced by calcium. Calcium is required for activating several proteins in the TCA cycle that then provide the power NADH for electron transfer and for ATP synthesis, but ultimately you can have ROS damage. And so these little green circles are for the calcium. And when you have damage, then what happens is that you get a breakdown of, as I just mentioned from Kulbrandt's study, you get a breakdown of this part that's forming the junction. And notice that the protein involved in this is the OPA1, which is also involved in the fission-fusion process. So when you have that, then the calcium rushes to the inside, and it's thought that the calcium will affect the dimer of the F1FO in some way to induce the transition pore. So here now, oh, I think I've jumped 2 slides. Yeah, let's go here. So I've been talking about the Podospora, lifespan of 18 days. Let's go to Drosophila, 50 days or more for the average lifespan of the cell. And what happens? In young flies, you have high oxygen consumption, low peroxide yield. And you can measure this by looking at the amount of catalase that is produced by the cell to consume the hydrogen peroxide. And there's also other enzymes that will try to get rid of the ROS that get formed. And so you can look at the expression levels of those. At the very beginning, you have lamellar, tightly bound lamellar cristae, and where you have a good bit of fenestration. But in the old flies, now what happens is that you have low oxygen consumption, high peroxide yield, and it's a clear correlation of the ROS-driven aging. And now you end up getting a very different structure here, where your cristae sort of circularize and they have big holes, and now then ultimately they fragment. They look they can be elongated, branched round. They can lack cristae altogether. Anyway, there's a clear correlation of how this aging occurs. Let's go to mice. So mice live about 117 weeks. Depends whether they're females or males. And so Kulbrandt did this study where he looked at young mice, 20 weeks versus the old ones at between 80 and 90 weeks. And he looked at 3 different tissues. So here we're looking at cardiac muscle. And he found that there wasn't that much change between the young and old, what he defined as old cardiac muscle. And he also measured the respiration and ATP synthesis and the amount of peroxide consumption. And he found that these cardiac mitochondria are protected from ROS somehow. He doesn't know how, but this kind of contradicts the ROS aging in cardiac cell hypothesis, where high respiration equals highest ROS, but then the high ROS gives rise to aging. Now, I'll tell you about a different study in just a minute that also extends this work. But let's look now at the, I think this is liver. I'm trying to read underneath my screen here at the top here. See if yes, this is the liver mitochondria. And so in the liver mitochondria, then you have 25% of the mitochondria are old, even from in the young ones, and they have large voids. And in fact, you'll even get these granular things that form in the middle of them, and where 80% are still in healthy things. But these are thought to contain the complex 4 and some oxidized lipids and calcium and glycoproteins. And so they're already looking rather sick. He also looked at mutator mice, where you're expressing this polymerase that has been altered so that you can generate lots of mutations of the mitochondrial DNA. And so you can see that you do still have laminar parallel cristae, but they also form voids as well. And so the mutator, both in the heart and even in the heart, you'll see this, but also in the liver, and you'll see that it really looks like it's old liver. Finally, the kidney. 80% of the young and old kidney, they look normal, but they're less tightly packed. But 20% of them, young and old, they resemble that of apoptotic cells. And so where there's no discernible pattern of cristae. So the different tissues, the mitochondria are aging and turning over at different rates, depending upon the tissue. And of course, with cardiac muscle, you really want to maintain that high healthy mitochondria to the best that you can, because your life depends on it. Okay, so they're subsequent to Werner Kuhlbrandt's studies, there has been this other study that came out where they also looked at the function of aging mitochondria with F1FO, and they found they went to much older, very close to the end of the lifespan of the mice, and so that they were able to capture in the heart mitochondria, where they could actually see the aging that occurred. And anyway, there is a lot of data here, but they did see the same aged mitochondria that Kuhlbrandt did not see in the cardiac, but he did see in the other tissues and in the other organisms, where he had the concentric ones. And at the same time, he was looking at ATP synthase, and he saw that as it aged, you lose ATP synthesis. And he was able to do some very sophisticated fluorescence tests where he could look at how much F1FO dimers he had versus monomers, and he saw that as you approached old age, that the amount of dimers of F1FO decreased, even though there was, and he saw a lower ATP synthase activity, as I said, even though there was no changes in the expression levels. There was still just as much F1FO as there was in the young one. So he's looking at this dicarbonyl stress that is a different way of degrading proteins. And how this works is that you have, you generate these dicarbonyls as a function of metabolism, and they can react, especially with arginine. And when they do, then they will degrade the functionality of a protein with age. And so what ends up happening is mitochondrial protein dysfunction increases. There's an increase in ROS, and it leads to activated apoptosis. So the glyoxyl and this methylated glyoxylase, they're maintainedly metabolized by this glyoxylase 1. So he went through and he did a very sophisticated experiment where he actually analyzed the extent that the F1FO gets glycated with age. And he would purify the F1FO, and he did a mass spec on all the peptide analysis, where he could actually measure the extent of glycolation and which subunits that it was in. And what he found was that it was mostly in these proteins that are specific for making the F1FO dimers. He then correlated that with the age and that the extent of glycation, and with the dimers and going into monomers by looking at these fluorescence diagrams. And so he shows a lot of very detailed data, and I probably should have broken this down into smaller bits, but again, he's looking at susceptibility of ROS. While Kulbrandt did not see this, then in fact, he didn't go with the cardiac muscle to, and he didn't get, he went to the mice that were in senior living, but were not yet at death's door. So by going to this more aged cells, then he found that he was seeing that ROS induced the permeability pore transition, and it shortened the secondary electron energetic collapse, and he could measure that by having these loaded cells that had a means to measure the amount of membrane potential that were inside them using MitoTracker. And he saw the collapse of the membrane potential. And then he also looked at the calcium retention, and he found that when cyclosporin A, and cyclosporin A is known to inhibit the MPTP. And so he was able to see an effect there as well. Yes, again, I got a little enthusiastic about this paper, so I wanted to share all the details. But anyway, he repeated this with these H9C2 cells, where he induced the dicarbonyl stress by on purpose, and gave him 3 days. And then he again repeated all these experiments where he was looking at the extent of dimer versus monomer F1FO, and saw that the increased glycation led to more monomers. And then ultimately the ATP synthase rates went down, and oxphos went down, ROS went up, and ultimately he was able to measure the MPTP formation as a result of the increase in calcium that he also followed in these cells. Now, Alistair mentioned that this is a rotary motor, the F1FO, and it uses both the proton gradient and the membrane potential to drive this rotation. My little animation here, the protons are coming through. They're at a non-equilibrium concentration gradient where they're higher on this side than they are on this side. As they come through, it drives rotation of the C ring relative to the stator, which is in the peripheral stalk, and also part of the F1 head that makes the ATP. So in response to that, you have 3 alpha beta heterodimers that each comprise in the F1 a catalytic site for ATP. And it acts like a 3-cylinder motor, where for every 120-degree rotation of the C ring, that there is a drive shaft that comes up in here. And as that rotates inside here, then you'll get 1 ATP driven, synthesized for at each catalytic site. So, and it will go around in a circle like a Wankel rotary motor. It's too bad they don't make these anymore and people are not that familiar with them, but it's very comparable to the F1. So while we know what the protons do, even though Mitchell got the Nobel Prize for this equation, where both membrane potential and proton gradients contribute to the ability to rotate and synthesize ATP, we still don't really know on a molecular basis what the membrane potential does, but clearly it is a contributor. In fact, it's the main contributor to synthesis of ATP in mammals in mitochondria. So, and we also don't know where this mitochondrial permeability transition pore occurs, but there's lots of people coming up with crazy ideas for it. One of them is up here at one of the ATP binding sites. It uses magnesium as a cofactor, and so people have thought, well, maybe it's substituting the magnesium for calcium, and the calcium is doing it. I'm not sure I buy the results that they've seen with that. Other people have said, well, it's causing a change in these proteins here that are forming this, and that's causing this to open up. And again, I don't see any evidence for it. In fact, it doesn't seem to form until you start to have ATP synthase monomers. So one of the third idea is that, oh yes, well, once it forms the monomers, now that changes the C ring, and now these molecules can rapidly go through the hole that's in the middle of the ring. The ring is filled with lipids, although those lipids don't show up much yet in most of the cryo-EM structures. But so again, I'm not buying that hypothesis. But this is one of the main things I think it's pretty well established that's going on with the transition pore with the ATP synthase, but exactly how it happens, I have a good idea, but I don't have any proof yet. So anyway, there's an awful lot of, I've talked about a lot of things having to do with aging, but one of the things is the mitochondrial DNA degradation that can occur. And the two genes in the F1FO are the ATP6 gene, the ATP8. The ATP8 is a little small accessory protein that helps dimerize F1FO. The ATP6, though, is subunit A, which is the critical protein in the stator that generates the torque on the C ring as the protons come through the C ring. It's known, and Doug Wallace has done an awful lot of research on this, that there are sensitive parts of the DNA sequence in the mitochondrial genome that are much more susceptible to mutation than other parts. And he's characterized a lot of these. And these are often, when you have mitochondrial diseases, which can be devastating, then these are the result of these. Well, here's a list of those mutations at specific locations within just these two proteins, the 6 and 8, the cytochrome A and this accessory protein. They start with the accessory protein. And you remember there's multiple copies of the mitochondrial DNA. So you can have heteroplasmy where some of them are still the normal sequence that is functional, but where you only have a few that are mutated. And it goes on. And if you look down here, the diseases, they're actually pretty bad. A lot of them are neurological. And so this is where they end up being located in the subunit A. And a lot of them are right at the interface to the C ring. So my work in the last few years, one of the major studies that we published just a couple of years ago had to do with what generates the torque of the protons traveling through subunit A as they go into the carboxyl group on the each C subunit in the C ring. The proton has to travel all the way around before it's then delivered back to subunit A, and where it goes to the cytoplasm. So it starts at the periplasm, comes in, goes out. Our work where we mutated several of these crucial residues on both sides here, we found that a lot of the torque can be generated by the proton dependent, translocation dependent step, which we found not to be move entirely the rotation of the C ring, but it would move it 11 degrees. So we did this in E. coli. The E. coli has a C10 ring, so it moves 36 degrees for each C subunit that moves a proton. But we found that the proton translocation event only moved at 11 degrees. So that was enough to take a proton that was here out in the lipid phase and move it in here. We could get energy derived from that, where once you get a negatively charged carboxyl, it wants to be excluded from the lipid phase. That'll move it the 11 degrees. So how does it get the rest of the way? Well, we think what happens is that once you make this negative charge, then that causes this attraction to this highly conserved arginine, and that pulls it the remaining 25 degrees so that the process can repeat. We proposed that because we could mutate all of these and we would change the pKa value of the entire thing, this had to be, these residues were not actually transferring the protons directly, but they were supporting a column, single column of water molecules that would go through, and these water molecules would actually hydrogen bond to the protons and move the protons through in a concerted manner by a Grotthuss mechanism. Subsequent to our coming up with this two-step mechanism, then cryo-EM structures have now gotten to the resolution where they've now been able to see the waters that we had predicted existed in this column, which was very encouraging. Okay, so the final thing is that where do all these pathological mutations from the DNA, how do they transform into this A subunit? They follow right along the pathway of the proton translocation pathway here. And so there's several more that occur. We can't explain all of them at this time, but we're working on it. So anyway, that's it, and I'll be glad to answer your questions.